Good morning everyone. Dr. Beena here. Today we are revising iron metabolism. In the revision we are mainly dealing with the university questions and the important questions were write briefly on absorption and storage of iron from intestine. Another was factors affecting iron absorption, and the third one was transport of iron. It can ask either way as absorption alone with factors affecting etc., and sometimes absorption and transport, sometimes storage, like that. And also one more question also can be asked. So in this diagram you can get an overall idea about the iron metabolism. Iron is we are getting iron through our food, and it is bound in the bound form and maybe in the ferric form. So iron is not absorbed in the ferric form. It has to be reduced to ferrous form by the ferri reductase, which is present on the brush border side of the intestinal mucosal cells. Then after that it is transported through the divalent metal ion transporter, and within the cell again it is converted to ferric, and that is by ferroxidase, and then it is stored in the form of ferritin, that is combining with apoferritin, and there it is stored as a ferritin. Then whenever there is requirement of the iron in the body, then it will be released, and it is released as ferrous iron, and then ferrous iron, and then it is passing through the basal membrane of the mucosal cell, and from the outside it is binding with transferrin, which is present in the blood protein, and then there again ferrous is converted to ferric by ferroxidase, which is which is the activity of ceruloplasmin present in the blood. It is a plasma protein, and then ferrous is converted to ferric, and then two ferric atoms will be combining with the transferrin and forms the transferrin, which is the transport form. Then it is taken to the concerned cell through their transferrin receptors, and within the cell the whole of the things internalized, and then iron is taken inside and the transferrin receptors are externalized. This is the overall summary of the iron metabolism. So we can go one by one. Before then, how it is distributed in the body, that is total content is 3 to 5 grams, out of which 75% is in the blood, and the rest is in the liver, bone marrow and muscles, and it is present almost all cells. So every cell contains iron. Then blood contains 14.5 grams of hemoglobin per 100 ml, and about 75% of the total iron is in the hemoglobin and 5% in the muscle, that is in the as myoglobin, or 15% as the storage form ferritin. Now what are the requirements of iron? Daily allowance of iron for an adult Indian is 20 milligram, out of which about 1 to 2 milligram only will be absorbed. That is only 10% of the total consumption is absorbed. And children between 13 and 15 years need 20 to 30 milligrams per day, and the pregnant women needs around 40 milligrams per day, because the requirement is more in pregnancy. Now what are the sources? It can be either vegetarian source or non-vegetarian. Vegetarian source mainly seen in the green leafy vegetables, and also pulses and cereals also. Pulses and cereals contains little less than the vegetables, but as an Indian culture we are consuming more of cereals, so we are getting iron through the cereals also. And non-vegetarian sources are, the main thing is the liver and meat, and they are rich in iron. Then jaggery is another good source of iron. So that is why in the children we used to give porridges added with where the jaggery is added. Then cooking in iron utensils will improve the iron content in the diet. So if you cook in the iron utensils, that will also improve the iron content. And another thing is the milk is a poor source of iron. Now we have to write about the factors influencing the iron absorption. Iron is absorbed in the upper part of the duodenum, and usually iron is absorbed in the reduced form. That is only in the ferrous form it is absorbed, not in the ferric form. Remember, only in the ferrous form. Then there are factors which increases the iron absorption, and there are factors which decreases iron absorption. So what are the factors increases? That is one important one is ascorbic acid or vitamin C, and that will help in reducing the ferric to ferrous with the help of gastric HCl, and then cysteine, amino acid cysteine, and the SH group of certain proteins. These are the most important factors which increases iron absorption, and those are substances which interfere with the iron absorption are phytic acid, which is present in cereals, and oxalic acid, which is present in the vegetables. Then there are some minerals which decreases iron absorption, that is calcium, copper, lead, and phosphate, and which will usually inhibit the iron absorption. One atom of lead will inhibit absorption of 1,000 atoms of iron usually. Now the absorption proper, it mainly takes place in the duodenum and jejunum, and iron metabolism is unique. That is particular that the homeostasis normal level is maintained by regulation at the level of absorption, not at the level of excretion. That is the importance. It is not absorption. Its level is not maintained by the level at the excretion, only at the level of absorption. When iron stores in the body is decreased or depleted, absorption is enhanced. When the adequate quantity of iron is stored, then the absorption is decreased. So whenever there is more need of iron, more absorption is occurring, and if there is enough iron in the body, the absorption is decreased. This is referred to as the mucosal block of regulation of iron absorption. Now the ferrous iron in the intestinal lumen, iron may be present in ferric form in the food, but it is reduced with the help of vitamin C or gastric HCl in the stomach, etc., to the ferrous form. Once the ferrous form is ferrous iron is formed, it binds to the mucosal cell protein called the divalent metal transporter 1, and then transported into the mucosal cell. So ferrous iron passes through the divalent metal transporter into the intestinal mucosal cell. Inside the mucosal cell, the ferric iron is formed and is complex with the apoferritin to form the ferritin. So after entering the intestinal mucosal cell, this again converted to ferric and then combines with apoferritin to form the ferritin, which is the storage form. The iron releases from the intestinal cell to the bloodstream. That is a separate method. The iron in the ferric form is released. Then it crosses the mucosal cell with the help of the transporter protein called the ferroportin, and the iron crosses the cell membrane and ferrous in the ferrous form. So again, from the ferritin, iron is released, which is in the ferric form. It is converted to ferrous, and then it is released through the ferroportin to the bloodstream. In the blood, it reoxidized to ferric state and then transported by the transferrin. That is, the transport form is the transferrin. So in the blood, the ferrous is again converted to ferric by the ceruloplasmin, which has got a ferroxidase activity, and it is converted to ferric. Then the ferric combines with apotransferrin to form the transferrin, which is the transport form. So that is the usual thing happening. So in this diagram, you can see the first is in the blood food, it is in the ferric form. Then here it is reduced to ferrous, and then the divalent metal transporter is there in spanning over the mesh border, and then through that it is reaching the mucosal cell, where it is again converted to ferrous. It is in the ferrous form. So then again it is converted to ferric, and ferric combines with the apoferritin to form the ferritin. So the whole thing is the ferritin, and now it is in the ferric form. Whenever iron is required, it is again this ferric form is converted to ferrous, and then it is released through the intestinal border, and it is combining with the apotransferrin to form the transferrin and transported through the blood. So you have to draw this diagram and explain whenever you are asked to write about the absorption. Now, how it is regulated? There are methods of regulation. One is mucosal regulation. That is, absorption of iron needs a divalent metal transporter and ferroportin. That is, ferroportin is from the mucosal cell to the blood, but the DMT1 is from the mucosal intestinal lumen to the mucosal cell. So the synthesis of both these proteins are down regulated by hepcidin. Hepcidin is a polypeptide which is synthesized in the liver when there is enough iron. So whenever there is enough iron in the body, hepcidin will produced, and hepcidin will down regulate the DMT1 and ferroportin. So what happens? The absorption will not take place. So if there is hypoxia or anemia, the synthesis of hepcidin will not occur. Only when there is enough iron, hepcidin is synthesized. So in the case of anemia, there is no synthesis of hepcidin. So if there is no hepcidin, there will be enough divalent metal transporter DMT1 as well as the ferroportin. So more iron can be absorbed through the transporter, and from the cell, more of iron can be released to the blood also. The hepcidin decreases the surface expression of the ferroportin, which is responsible for the moving of iron that we have already explained. Now the second method of regulation is the stores regulation. As the body stores are fall, that is iron content is falling, the mucosa is signals to increase the absorption. Similarly, in erythropoietic regulation also is there. In response to anemia, erythropoiesis means the production of RBC. So in response to anemia, the erythroid cells will signal the mucosa to increase the iron absorption. So when there is anemia, the mucosal cells are signaled to increase the absorption. There is a reciprocal relationship between the synthesis of ferritin and transferrin receptors. That is, when there is enough more iron is there, then more of ferritin is stored, but that time there is no requirement for the cell. So that is why transferrin receptors are not produced, and there is no uptake into the cell. Then the transport form of iron is transferrin. We have explained how it is forming in the blood. Ferrous is taken to the blood, where it is converted to oxidized to ferric by the ceruloplasmin or peroxidase activity. The protein ceruloplasmin to ferric form, then it combines with apotransferrin to form the transferrin. So the mode of transferring iron in the blood is the transferrin form. Don't get confused. Ferritin is the storage form, transferrin is the transport form. Normal plasma level is 250 milligram per 100 ml. In iron deficiency anemia, it is increased, and 1 molecule of transferrin can combine with 2 atoms of iron, that is in the ferric form. 2 ferric form combines with 1 apotransferrin to form the transferrin. Then the TIBC, what is TIBC? Total iron binding capacity in the plasma, that is 400 microgram per 100 ml is the value. This is provided by the transferrin again. 1/3 of the capacity is saturated with iron. In iron deficiency anemia, the TIBC is increased, that is level is increased, but the serum level is, serum iron level will be decreased, but the TIBC level will be increased in anemia. In blood, ceruloplasmin is the peroxidase, which you oxidize, that we have already explained how it is oxidized in the blood, that is by the ceruloplasmin. And there are transferrin receptor, that also we have explained, that is taking the iron transferrin complex into the cell, and the whole the complex together with the transferrin receptors are taken to the cell, where it is internalized, and iron is entering the cell for the use. So iron transferrin receptors are mainly seen on the cell membrane, where the hemoglobin synthesis occurs. So the iron transferrin receptors internalized, and iron is taken by the cells. Now the few words about the storage. There are 2 forms of storage. Mainly the one is the ferritin. It is the storage form seen in an intestinal mucosal cell, liver, spleen, and the bone marrow. The apoferritin can take to 4,000 iron atom per molecule. The ferritin contains about 23% of iron. So the major part of the storage is in the form of ferritin, where around 4,000 iron atom can be taken up by an apoferritin. Normal plasma contains very little ferritin, but ferritin in the plasma is increased in iron overload. When there is excess iron in the body, ferritin level will be increased, and the ferritin level of the blood is an index of the iron status of the body. Another synthesis of transferrin receptors and ferritin are reciprocally controlled, that we explain. When there is enough iron, the iron is stored in the ferritin form, then that time there is no requirement in the cell, so there won't be transferrin receptors produced, so there won't be intake into the cell because excess is stored as the ferritin. In iron deficiency anemia, ferritin content is reduced. Then another storage form is the hemosiderin, that is formed by the partial deproteinization of ferritin by the lysosome and are found in aggregate in tissues like liver, spleen and bone marrow. So again, this is a partial deproteinized type of storage form of iron, that is from the ferritin it can be produced, and it is more insoluble. It is not soluble as the ferritin, and the iron is more slowly released. So it is not soluble, so that the release of iron is very slow from the hemosiderin. So when you are asked to write about the storage form of iron, you can write the ferritin mainly, and also you can mention hemosiderin. It's a partially deproteinized form of ferritin which is also can be considered as a storage form. So this much regarding the iron metabolism for you, mainly the absorption. Sometimes to sources also can be asked. So vegetarian and non-vegetarian source you can write, and the RDA recommended allowance, that is 20 milligram per day, and the method of absorption you can draw the diagram and explain. And then a transport form of iron is transferrin, and the storage form of iron is ferritin. Thank you.